Hello and welcome back to Excelsior Academy. So today we are going to do the fourth part of IIT JAM 2019 Biotechnology, solving with the explanation. So I hope this series is being useful for you guys. If still any doubts are there, please comment, and you can also message me into the Telegram by the name Excelsior Academy. So let's do our fourth series. So 31st question is, pick the correct statements with respect to the interconversion of the topoisomers of a circularly closed double-stranded DNA. Now, this is very simple. If you have gone through my lectures of biochemistry series, mole bio series, then over there by DNA topoisomerase, I have taken a lecture. So if you have seen that video, then very beautifully you can answer this question. Okay, so let's see what are the options given. Only one strand needs to be cut. Actually, this is true. If you remember DNA topoisomers for that of a circularly closed double-stranded DNA, we know that there are two types of isomerase in order to convert the topoisomers of the DNA. So we have got the type 1, which needs to break one strand of the DNA, and then we have got the type 2 also. So from this, it is very clear that it is going to break 2. So only one strand needs to be cut. It is also there, right? Both strands have to be cut. Yes, as I mentioned just now, no strand needs to be cut. This is wrong because in order to interconvert or change any of the topoisomers of a DNA, then it is required that there has to be a nick in the DNA. Now, ATP is required for interconversion. Yes, it is true because type 2 topoisomerase requires ATP in order to break the strand. So hence, for this session, you need to select all the correct statements. So over here, the answer is A, B, and D. Okay, so I hope this is clear. If it is not, please go to the lecture by the name DNA topoisomers. Over there, it is very nicely with the diagrams. It has been explained fully. So moving on to the next question. Now this is mathematical question. So let U is the subset, it's having the elements of 1 to 15. Let P is a subset of U having all the prime numbers, and Q is a subset of U consisting of all the even numbers, and R is a subset of U having all the multiples of 3. So this is very simple. This you have to write like as. So P is equal to, you can write like as it is given that it is all the prime numbers. So from 1 to 15, all the prime numbers you need to write. So 2, 3, 5, 7, 11, and 13. Right. So how many elements are here now? All the prime numbers are there, and we have got these 6 elements. Now Q is all your even numbers from 1 to 15 you need to write. So 2, 4, 6, 8, 10, 12, and 14. Okay, how many are there? Again, 7 elements are there. Next is R, which consists of multiples of 3. So R is multiples of 3, 3, 6, 9, 12, and 15. So how many are there? Over here, 5 elements are there. Okay. Now, next is let T is equal to P minus Q. So now you have to find out what is T is equal to P minus Q. So that is P minus Q. So what you're going to get? So when you solve it, you're going to get as 3, 5, 7, 11, and 13. So how many are there over here? You have got the 5 elements. So your T is 5. So now we see that there are two options, A and C, having T as 5 and 5. Right? Now, okay, so these two are might be correct answer. Now, which of the following statements are true? Now, we need to calculate whether these are correct or not. Okay, so for this, T union R, you need to calculate. So that is your 3, 5, 6, 7, 9, 11, 12, 13, and 15. Okay. So this you are going to have 9. So I have already solved and kept. That's why you can see whenever we are writing, as already mentioned, what is T, R, and P. You can solve it over there. So now we have got 9. So from this we know that T union R, how many are there? 9. So yes, this statement is correct. Now let us talk about the T intersection R. Okay. Now for this, we are going to calculate T intersection R. So what is T intersection R? So we are going to get only 3. Okay. So it means what? How many elements are there? How many elements are there? Only 1 is there. So it means what? The second statement, that the C is also true. Hence, the answer is that A and C both statements are correct answer for this particular question. So I hope this is again clear to you guys. This is very simple actually. I hope this is clear to all of you. Moving on to the next question. Now let f of x is x minus 1, x minus 2, x minus 3, x minus 4, and let alpha is f of 3 by 2, beta is f of 5 by 2, and gamma is f of 7 by 2. Now again, which of the following are the correct statement? Now keeping this into the mind, we need to calculate whether it is having the same sign or not. Okay, so when you talk about same sign, it means they're just asking whether both alpha, beta, gamma are having the positive sign or the negative sign. So when you put these values, you just need to know whether they are giving you the positive sign or the negative sign. Okay, now for first case, so in the first case, f of x, your x is greater than, oh sorry, for f of x, your f of x is positive if it is positive only when you have got x less than 1, right? Then you have second condition when 2 is in between of, sorry, x is between the 2 and 3. The third condition is when you have x greater than 4. Okay, so from these factors you can actually identify your f of x is going to be positive only when these conditions are satisfied. Now let's see what are the alpha, beta and gamma sign. Okay, so in the first case, when x is over here less than 1, we see that alpha condition where alpha, when alpha is f 3 by 2, that time when you put these values, you see that it is going to be negative. You just put the value and see, you are going to get the negative answers. Okay, so in the case of alpha, it is going to be negative. Now when you put beta, you see that these values are falling in this range. Okay, so it means it is going to be positive. Now let's check about the gamma also. Put the value and you see that it is falling between the 3 3 is greater than x is greater than 4, and now then this value is going to be negative. So now what we got is that alpha and gamma is going to have the same sign, that is negative negative. Okay, now let's see what are the options are there. So alpha gamma have the same sign. Yes, this is the correct option. So here, what about the other? Alpha beta and beta gamma have the same sign. So if you multiply alpha beta, negative positive, you're going to get negative, and when you multiply beta gamma, again you're going to get negative. So again the fourth statement where alpha beta beta gamma have the same sign, so it is correct. So for this question the answer is B and D. Okay, so it's very simple, just put these values into the equation and then solve it. Now the next question is the characteristics oxygen binding profile of hemoglobin is shown below arises due to the following. So let's see what are the options are given. So quaternary structure. So many a times whenever I was teaching mole bio as well as biochemistry, I have said that the best example for quaternary structure is hemoglobin. So yes, the quaternary structure is there for the hemoglobin, right? So so now hemoglobin is having 4 subunits of the globin. Okay, now each with having each subunits. So here your hemoglobin has got the 4 subunits, and each subunit, for example, let us say that this is your hemoglobin. Each subunit is having the heme group, and these heme group is having your Fe. Okay, so iron is the main component that actually binds to the oxygen, and each hemoglobin molecule is able to carry 4 molecules of oxygen. So each each hemoglobin molecule is able to carry 4 oxygen molecules. So even this is correct. Okay, now talking about the subunit dissociation. So actually in case of hemoglobin, whenever one subunit binds to oxygen, okay, let's say that this is your hemoglobin. The moment it binds to oxygen, it is helping the other subunits also to get bind to the oxygen. Now this is called cooperativity. So even this is true, but it cannot be subunit dissociation, because when the subunit gets dissociated, the activity falls actually. But here we see that it is increasing, so it is not. So the subunit dissociation of hemoglobin does not contribute in arising of the oxygen binding profile. Okay, actually dissociation will result in the opposite way. That is, the binding capacity of oxygen is going to get reduced that time. and cooperativity, just now I have discussed. Now the last one is conformational change. So the bonding of the hemoglobin molecule with the oxygen changes the conformation of the binding site. Now this will increase the affinity for the other oxygen molecules to bind the hemoglobin protein, right? So now we have got the hemoglobin in two conformation. So always I say that open or close. So in the same way, hemoglobin is also going to have in the two conformations. Okay. So if you remember your GLUT transporters over there also we have talked about open and close, or sometimes we have also mentioned that it is in the tense form or the relaxed form. In the same way, your hemoglobin is also having two conformation, that is T form and the R, tensed and the relaxed form. Okay, so these are the two conformation. So oxygen binds with much higher affinity in the R state. So higher affinity in the R state, and T state is a conformation of deoxyhemoglobin. So this is your deoxyhemoglobin conformation. Now the binding of oxygen to the hemoglobin subunit in the T state triggers. So what is happening? So your hemoglobin is initially in the T state. The moment oxygen is binding, it is getting converted to the R state. Okay, so yes, conformational is also the answer. So here the options are A, C, and D. Okay, not subunit dissociation, quaternary, cooperativity, and conformational change. So your answer is A, C, D over here. Okay, so moving on to the next question. The advantage of storing chemical energy in the form of starch and not as free glucose are that it dash. Okay, so this is again very it's a very beautiful concept, like why we don't store our energy in the form of free glucose, because that is the instant source of energy. So you know that whenever someone is fainting and also we immediately give the glucose drink to that person, right? In the same way, in the case of plants also, the instant energy source is glucose, but whenever we want to store our energy, we don't store in the form of glucose. We store it either in the form of starch or in the form of the glycogen in the case of animals. Now, in the case of plants, it is stored in the form of starch. Why? Because, see, the thing is that when the glucose are converted into starch, it becomes insoluble in water, and then it cannot be transported to the other plant cells unless until it is converted to glucose. So now this is minimizing the diffusion. Okay, so it is correct. So it minimizes the diffusion, because if it is present in the glucose, whenever any cell is present, because of the osmotic potential difference, it will be then moving from one place to another. So you cannot store the energy in the form of glucose. It will be used, it will be readily used up. So that's why we are keeping in the form of starch in order to minimize the diffusion. Now, starch also consists of two components. Amylose is a, amylopectin is a. Now, these structures enable the starch molecules to coil into the compact storage. So, yes, it is also enabling to go for the compact storage, as just now said. Now, since starch is insoluble in water, but its presence means that there is a solute potential which decreases the water potential. So, this also we know that if you know the water potential, it is because of the solute, gravity, as well as the osmotic, right? So once, even though we say that starch is present, it is insoluble in water, but it itself is acting as a solute, right? Solute molecule. Now whenever these solute molecules are present, what will happen? The water potential will decrease, and hence it reduces the osmotic pressure. So even this statement is also true. Now, when conformation of open chain glucose is converted to cyclic glucose, carbon number 5 will react with the aldehyde group and form the cyclic conformation. Now, this aldehyde group loses its reactivity because it gets bind with the carbon. So, it protects against the chemical reactivity of aldehydes. Okay. Yes. See, if you have got glucose, like in the open structure, it has got many open reducing ends are there. But once it is in its cyclic form, it actually protects the chemical reactivity of the aldehyde group. So that's why even this statement is true in the case of this question. So I hope this was helpful. So let's move on to the next question. So which of the following cell types can develop from myeloid lineage? So for this question, actually, again, very simple. If you have seen, if you have gone through the animal tissue culture techniques lecture over there in the stem cell technology, I have taken the lineage of the myeloid. So the answer is very direct from that. It's macrophages and the erythrocytes. Okay. So from a common myeloid progenitor cell, both erythrocytes and macrophages are arising. T lymphocytes and B lymphocytes, they arise from the common lymphoid progenitor. Okay, so myeloid will give you macrophages and erythrocytes, whereas lymphoid lineage will give you T lymphocytes and the B lymphocytes. So again, just for extra information, just let me share this. Okay, so let me share. So macrophages and erythrocytes are from the myeloid, whereas these two are from the lymphoid progenitor. Now, so for common myeloid progenitor, you get is erythrocyte, mast cells, thrombocytes, and myeloblast. Okay. Now, from the myeloblast, you get your basophil, neutrophil, eosinophil, and monocytes. Now, from monocytes, you get on maturation macrophages. Okay. So this is about the myeloid lineages. Now coming to the lymphoid progenitor, like from the lymphoid progenitor. So you guys can also make such flow chart so that it becomes easy for you guys to remember. You get your natural killer cells and lymphocytes. Now from lymphocytes, obviously you get T and B lymphocytes. And from B lymphocytes also you get the plasma cell. Okay. Yes. So let's move on to the next question. Electromagnetic waves dash. So whether they carry energy, whether they carry momentum, whether they are transverse in nature while traveling in vacuum, or they do not need a material medium to travel. So for this, we need to see all the options. So in this case, electromagnetic waves, they carry energy, and this is very obvious, right? So these energy they carried by the electromagnetic waves, it is inversely proportional to its wavelength. So this also we know. And even if you're not that much thorough in your physics, but still this you should know that, and it is equal to your h by lambda. That is, this is your wavelength, or other is also called h nu, that is your frequency, where h is the Planck's constant. At least this much should be known even if you don't like physics to study. Now electromagnetic waves carry momentum which is proportional to energy. So this is given as E by C, where C is the speed at which electromagnetic waves travel in vacuum. So it is also known as speed of light. So I hope you know what is speed of light, what is the value. Okay, please remember, need not to be said again and again. And electromagnetic waves, they are transverse in nature while traveling in vacuum or free space, that the changing magnetic field produces electric field and vice versa. So even this is correct. A, B, C, correct. Now, electromagnetic waves do not require a medium to propagate because medium is required by the mechanical waves and not with the electromagnetic. So even this is true. So for this question, all the options A, B, C, D are true. Now, which of the following statement is or true? So in intrinsic semiconductors, the number of electron is equal to the number of holes at any temperature. So yes, this is true actually, because the number of holes in the valence band, it is equal to the number of electrons in the conduction band. So as the temperature is increasing, so these free electrons and holes gets generated equally. So the number of electrons leaving the holes, so obviously those many numbers of holes will be present in the band. Now, an intrinsic semiconductor changes to an n-type semiconductor upon addition of a trivalent element. Now, when an intrinsic semiconductor is doped with the trivalent element, it becomes actually a p-type and not the n-type semiconductor. Because in p-type, it means that the semiconductor is rich in the holes. okay, or the positive charge ions. Whereas when intrinsic semiconductor is doped with the pentavalent element, then it becomes n-type semiconductor. Okay, so your here option A is correct, but not your B option. The shape of the IV characteristics of PN diode are curved or curved shape and not the straight line. So again, the statement C is wrong. Now, PN junction diode allows the minority charge carriers. So positive charge carriers with cross the, it will cross the PN junction. They are attached towards the negative terminal and the negative charge carriers attracted towards the positive terminal. So PN junction diode completely blocks the majority charge carrier. So hence this statement is true. So option A and D is the correct statement for this particular question. Okay. Now, coming to the next. So BF3 is also called a boron halides. They are actually the electron deficient. Okay. So they are just asking to which it is going to react readily. Now, since they are the electron deficient molecules, they do not have the complete octet of electrons surrounding the boron atom. So they tend to act as the Lewis acids since they are the electron deficient. So they tend to act as Lewis acids by accepting the electron pairs from the bases to form the stable acid-base adides. Now such donors will be from the pyridine or the pyridine stainless chloride complex. So it will react more readily with the C5H5N or the SnCl2. Okay. So your option A and D is the correct answer over here. Now the last question for this session. The reaction of 2-bromobutane with Cn minus proceeds by dash. So reaction proceeded. So this reaction will proceed by the inversion of the configuration over here. So if reaction is started with the R2-bromobutane, and then reaction will end up with the S2-methyl butane nitrile or the vice versa. So that can be written as Br. This is 1, 2, this is 3, and this is 4th carbon. Okay. Now when you have got Cn minus, you are going to get as 4, 3, 2, 1. This will be over here. Okay. And what you get is NaBr. So Na plus Br minus you're going to get. Hence, based on this, it is the retention of the it is the inversion of the configuration as well as formation of the new product. So hence, this is the answer. So for this little bit of organic chemistry is needed. It's not like you need to know the entire thing. You can just remember the important organic reactions which will satisfy, which will be enough for you to remember and to crack the exam for the IIT JAM. So that's all for today, and we'll come back again with the 5th section of this particular series. If any doubts are there, please comment, or you can ask me in the Telegram also. So keep studying, and yes, all the best for your future endeavors. Thank you.